Okay, so now I'd like to thank in advance Dr. Anthony McGoron for giving this talk today. And please, Professor Elcio, I would like to ask you to introduce Dr. Anthony.
Oh, good morning, all. It's a great honor for me to introduce you today. For today's seminar, Dr. Anthony McGoron. He's a professor of biomedical engineering, and he's the Associate Dean for Academic Affairs at the College of Engineering and Computing at Florida International University. He received his PhD in biomedical engineering from Louisiana Tech University. His postdoctoral training in the Department of Pharmacology and Cell Biophysics at the University of Cincinnati. Before joining FIU, Dr. McGoron was an assistant professor at the University of Cincinnati, Department of Radiology, Divisions of Nuclear Medicine and Medical Physics. He's a fellow of the American Institute for Medical and Biological Engineering. He has also served as interim chair of the Department of Biomedical Engineering in FIU from 2007 to 2010. And he was the national president of the Alpha Eta Mu Beta Biomedical Engineering Honor Society from 2010 to 2014. He has over 200 journal articles, book chapters, and proceeding papers. And he has been funded by different governmental agencies like NIH, NSF, DOD, as well as from different companies. And Professor McGoron is also the editor-in-chief of the journal Critical Reviews in Biomedical Engineering. So without further delays, let me thank Professor McGoron for presenting this seminar today. Let me pass the screen, the floor, the word to him. Thank you, Anthony. Thank you.
All right. Well, thank you very much for the introduction. I want to make sure you're looking at the right screen here for my presentation.
Yes. Yes.
Okay. It's perfect.
Great. So again, thank you for inviting me to give this talk today about nanoparticle drug delivery in cancer. I want to go over a little bit of data and really kind of have a discussion about why haven't we found the silver bullet with nanoparticles for cancer therapy. Before I start, I want to, if you invite anyone to, if they make it to Miami, to come visit the campus. We have a beautiful campus. FIU is the 4th largest university in the United States. If from this talk, anybody finds some interest and value, I welcome you to contact me, and maybe we can start up some discussions about how we can move some of this technology further. I want to remind you all, I'm an engineer, not an oncobiologist, so you might have to forgive me for some of the biology that I present. But let's start off with a little history first. Liposomes were discovered or maybe invented in 1965, and liposomes became the foundation for nanoparticle drug delivery. It was about 15 years before the ability to get them to the small nanometer size and to label them with polyethylene glycol, which is a polymer, a neutrally charged polymer that makes the particles stealth in the blood, makes them invisible, avoid opsonization of plasma proteins, and therefore extend plasma half-life. And then another 15 years before the approval of the first nanoparticle drug delivery system, liposomal doxorubicin called Doxil, and then other particles, nanoparticle drug delivery systems for cancer that were developed later. There are many different types of nanoparticles that can be used. For instance, nanocrystals of the drug itself, stabilized by a polymer. The liposomes, which I'm sure that you know is a bilipid membrane with a polar head sticking towards water on the outside and inside, and a lipid tail. And hydrophilic drugs can be delivered in the interior of the liposomes, or hydrophobic drugs in the lipid membrane part. Other types of particles, polymeric micelles or protein-based nanoparticles, for instance, abraxane, paclitaxel protein construct, dendrimers, carbon nanotubes, and of course, conjugated drug polymers, depending on their size, may not technically be classified as nanoparticles. Usually when we're talking about nanoparticles, we're talking about in the 50 to 150 nanometer range. So what's the motivation for delivering drugs by nanocarriers or polymer conjugates? First, to improve the solubility of the poorly water-soluble drugs, reduce drug metabolism, increase the plasma resonance time by making them stealth, reduce kidney excretion, the ability to deliver high payloads directly to the target, and the ability, at least potentially, to add imaging functionality to without affecting the pharmacodynamics, and therefore creating a theranostic. The potential advantage of mesoporous silica nanoparticles, which I'm going to talk in a little bit of detail about, is that they have potential for higher loading capacity to the high porosity and surface area, a wider array of drugs that can be incorporated compared to liposomes or polymer nanoparticles, greater delivery of the release of the drug, potentially greater stability in the circulation, prolonged drug release, which may or may not be beneficial, the inherent ability to release the drug in a low pH environment of the cancer tumor environment, or attach thiol cleavage of the drug to be released in a high glutathione tumor environment, which is often found in many cancers, and greater flexibility for active targeting in the delivery of multiple drugs simultaneously. So mesosilica particles, which I'm very interested in, although I've also investigated other types of particles that I'll briefly talk about, but mesoporous silica particles start with a surfactant, and the surfactant creates a micelle, and these micelles under optimal conditions can produce rods or other arrangements, add a silica precursor to it, and silica polymerizes and condenses around the micelles, and then when the surfactant is removed, results in some very ordered porosity in these nanoparticles, and the drugs can be delivered in these pores. Now, what would the ideal drug delivery vehicle look like? Well, it might be either a polymer or a metal, have an imaging element to it if it's an imaging agent or a theranostic. The imaging could be iron oxide for MRI imaging, or near-infrared dye for fluorescence imaging, or a PET or a SPECT isotope for nuclear medicine imaging. Combined with a therapy, which could be chemotherapy or maybe proteins or DNA, would have a stabilizer that makes it stealth, have a targeting moiety. So next I want to switch gears to hyperthermia, because the interest in combining chemotherapy with hyperthermia. So this is just from the cancer.gov NIH website. Hyperthermia is a type of the treatment which the body tissue is heated to help damage and kill cells without harming normal cells. It can be induced by microwaves, radio waves, lasers, ultrasound, or heating the body, or fluids that are administered to the body. And hyperthermia is widely available, and it's been used to treat many different types of cancers. It can be delivered through a nanoparticle formulation using magnetic nanoparticles, which can be induced to heat in a magnetic field, gold nanoparticles, which can be induced to heat in a near-infrared irradiation, carbon nanotubes, which also would respond to near-infrared radiation to increase temperature, and also different types of nanoparticles that can encapsulate near-infrared absorbing dyes. For instance, polymer nanoparticles or Mesoporous nanoparticles. Hyperthermia can be delivered either globally or regionally, which in the next few slides I'm going to refer to as a slow rate induction of hyperthermia, or a localized hyperthermia, and in particular by the introduction of a drug which can be then sequestered in the tumor, travel to the tumor, be sequestered in the tumor, and then be irradiated with some external energy to excite the particle to produce heat. And this type of introduction of hyperthermia I'll refer to as a fast rate hyperthermia. And this is primarily what I'm interested in inducing hyperthermia with nanoparticles. So here I show some data using a drug, indocyanine green, which many of you may be familiar with, but it is an FDA approved, Federal Drug Administration in the US approved compound for measuring blood flow because it binds with plasma proteins. But it also is fluorescent in the near-infrared range, and it also absorbs the near-infrared light and can produce heat. So here I show when we expose a 2.5 or 5 or 10 micromolar concentration of indocyanine green to an 808 nanometer laser at 6.7 watts per square centimeters, the ICG will absorb the energy, and part of the released energy is fluorescence, but part of the released energy is heat. And with a 5 micromolar concentration, we can increase the heat to 43 degrees. 43 degrees is what I refer to as a mild hyperthermia. It's not enough to ablate the cells, but it's enough to modify the biology of the cells, and cancer cells respond to this mild hyperthermia at about 43 degrees, whereas normal cells do not. So this 43 degrees is our target for our, what I call mild hyperthermia. If we place the same volume of solution in an incubator, in a cell culture incubator at 43 degrees, it takes about 1 hour to reach the 43 degree mark, whereas with the laser-induced hyperthermia, it occurs in about 1 minute, 60 seconds. So this time is seconds, this time is minutes. So 60 seconds to increase the environment to 43 degrees versus 1 hour to increase the environment to 43 degrees. So again, this is my model for a fast, localized hyperthermia, and this is my model for a more global, slower rate hyperthermia. Now, of course, we need to show that the laser itself doesn't induce damage to the cells. So we grew cells and exposed it to the same laser with the same energy fluence, and showed that even after 5 minutes of exposure, there was no effect on the cells. Here we looked at the combination of hyperthermia and chemotherapy. This is a study that's more than 10 years old now that my graduate student did, in which we exposed cells to different treatments and at different concentrations of doxorubicin, which I'm sure you're familiar with as a chemotherapy agent, which had the expected dose response. If we add doxorubicin with 5 micromolar indocyanine green, the indocyanine green doesn't have any effect on the cells. If we put the doxorubicin, put the cells in an incubator for this slow rate hyperthermia, we see the slow rate hyperthermia has some effect, and then adding the doxorubicin has an additional effect. But if we do the same experiment by exposing the cells to the doxorubicin, the indocyanine green, and 1 minute of the laser, which I showed before, increases the temperature quickly up to 43 degrees, we have a much greater response. So we see some synergy between the doxorubicin and the fast rate hyperthermia, but not a synergy between the doxorubicin and the slow rate hyperthermia. So I'm very interested in learning more about what's going on at the cellular biological level as a response to these different types of induced hyperthermia. Someone watching might have some ideas of how we could pursue this further. We also looked at delivering our drug with nanoparticles. Here, I'm using PLGA nanoparticles, polymer nanoparticles, and investigating 3 different cell lines. SCOV3 is a human ovarian cancer cell line that is chemo-resistant due to a caspase-3 mutation. And so these cells don't easily undergo normal apoptosis. Mesocells, which are a human uterine cancer cell line, which are highly sensitive to chemotherapy, and DX5, also referred to as MESA and DX5, which are the human uterine cancer cell line, but with upregulated P-glycoprotein for multi-drug resistance. So these DX5 cells can efflux the doxorubicin and similar types of drugs to help protect the cell. What we showed here is that the uptake of the indocyanine green and doxorubicin delivered by PLGA nanoparticles, there's no difference in the SCOV cells because they don't resist uptake of the free drug. There's no difference in the mesocells, but we see a clear difference in the DX5 cells. The presence of the P-glycoprotein keeps out the doxorubicin, but by delivering it through the nanoparticles, we can get the drug into the cell. Now, we did this is kind of a complicated slide, but we did another study with the same 3 cell lines in which we added a drug, verapamil. Verapamil is an inhibitor of P-glycoprotein function. And then also the free drug and the free doxorubicin. The third is verapamil with the free drug and free indocyanine green, the free doxorubicin and indocyanine green. Then with the 2 drugs delivered with the nanoparticles without laser, delivered with the nanoparticles with 3 minutes laser. Excuse me? Okay, 3 minutes of the exposure to the nanoparticles with 5 minutes of laser, and then also with the verapamil. We see an expected response in the SCOV3 cells, and the mesocells, it's easy to kill these cells. They're very sensitive to chemotherapy. But with the DX5 cells, we see that delivering the drug by nanoparticles to multi-drug resistant cell lines, we have, first of all, we could increase the effect of free drug by delivering verapamil, and that's been tested and even studied clinically, although I don't think it's ever been made practical, and so it's really not an option clinically. But we could increase the uptake, and that would be terrific. But with nanoparticles, we can increase the uptake, but we also get a much greater effect if we also induce the hyperthermia. Here I show that the verapamil doesn't really add too much extra in terms of verapamil getting more of the drug into the cells because the nanoparticles are already bypassing the P-glycoprotein, so they don't need that extra boost. So here I'm just going to show one slide that relates to what might be happening on the oncobiology side of the of this fast rate hyperthermia. So what we did is we took the chemosensitive cell line, MESA and in the chemo-resistant cell line, the MESA DX5. And we exposed them to a 43-degree incubator hyperthermia over 1 hour, or the indocyanine green induced hyperthermia over 3 minutes. And we see that in the incubator hyperthermia, there's an upregulation of heat shock protein 70, and which doesn't occur in the slow rate hyperthermia. So we believe that that the slow rate hyperthermia might give the cells time to upregulate their heat shock protein and therefore give some protection to the cells. The same study, this was a study in 2013, also by my graduate student. And we do have a lot of other data in that paper showing that the localized hyperthermia primarily induces necrosis, whereas the slower rate hyperthermia primarily induces apoptosis. But I don't really have time to show that because I want to get to my larger point, which is what do we need to do to make nanoparticle drug delivery more effective? So what is the problem here? I've shown positive data showing that nanoparticles seem to work very well, but what is the problem? Well, the problem is that the particles have to take a very difficult route between where the drug is injected into the body until it gets to its target. So first it has to evade capture by the Kupffer cells of the liver and the spleen, and also circulating macrophages. And this is primarily done by making the particles stealth. That is, making the particles, forming a polymer around the particles, and also reducing the charge on the particles to near neutral. The size of the particles need to be large enough that they escape glomerular filtration in the kidneys, but they need to be small enough to escape the capillaries and get into the tissue. And many cancer cells express a phenotype of enhanced permeability and retention. Many cancers, but not all cancers, have clefts between their endothelial cells because the vasculature is not well developed. It's immature. And so there are holes between the endothelial cells that aren't found in normal tissue. So this allows these nanoparticles that are that size or smaller to be able to get out into the tissue. So the particles have to be smaller than these clefts, but they have to be larger than the glomerular filtration, which is roughly 5-10 nanometers. To get through these clefts, they should be 100 nanometers, maybe 150 nanometers. But once they get into the tissue, they have to diffuse throughout the tissue to actually reach the cells. Once they reach the cells, they have to interact with the cell membrane and get into the cells. Once inside the cells, they have to release their drug to get to whatever the site of action is. Now, that's assuming that the target is inside the cell, which may not necessarily be the case. And I'm going to talk about that in a couple of minutes also. And also, keep in mind, I'm sure that this audience is very familiar with the biology of cancer and understand that tumors, the tumor microenvironment is complicated and crowded. There are lots of different types of cells within the tumor microenvironment. The center of the core of the tumor tends to be hypoxic, tends to have lower pH. And so we really have to account for all this when we're dealing with the transport of the drug to its target site. Another big problem, of course, is the cost to create new drugs. This is a plot of the 10-year R&D spending in billions versus the number of drugs approved. And the cost is very high. The cost depends mostly on what it costs to conduct studies to prove it is safe and effective and secure regulatory approval, from $10 million to $2 billion per drug. 90% of medicines that start being tested in people don't reach the market because they're unsafe or ineffective. Now, the 2.7 billion figure that they came up with in this paper includes the cost not only of the failures of the drug, but also of not putting money into drugs that might have been successful because you spent the money on the failures. Now here, I'm going to show a few slides that are a little bit sobering reality of the pegylated liposomal doxorubicin, or liposomal doxorubicin without pegylation. So here's a meta-analysis study and shows a forest plot of the overall survival according to whether whether it was a first-line therapy or a second-line therapy compared to a traditional therapy, whether it is a non-pegylated or a pegylated liposomal formulation, and then the effect size. And this shows the odds ratio, which a odds ratio of 1 translates to no effect. So if we look through all these data, we see that all of these effects overlap 1, meaning that there is no overall survival advantage to these treatments. If we look at progression-free survival, again, only a couple of the studies are outside the odds ratio of 1, meaning the effect is really rather small. Response rate, very similar if we look at the data across different studies for this response rate. So the conclusion here is that liposomal doxorubicin containing different chemo regimens do not improve overall survival compared with other schedules, while a marginal progression-free survival advantage is observed in these schedules. So this certainly doesn't mean that there's no advantage to liposomal doxorubicin. It has been a commercial success, but of course, more importantly, it has been a success in the clinic by improving quality of life and reducing off-site toxicities. In particular, it reduces cardiac toxicity, which is a huge problem for chemotherapy patients, particularly for doxorubicin, because the dose of doxorubicin the lifetime dose of doxorubicin is limited to a certain amount because of primarily because of cardiotoxicity. And the doxorubicin, while being very cheap, as we know, most cancer patients don't really die necessarily from the cancer. They die from the recurrence of the cancer because we're unable to continue the treatment. So here, I'm not going to go through all of this in detail, but there are several other meta-analyses that really all came to the same conclusion. This meta-analysis is for different cancer types: ovarian cancer, another one for ovarian cancer, breast cancer, melanoma, all really coming to the same conclusion that there is little, if any, overall survival benefit to the liposomal doxorubicin, although there is reduced side effects and toxicities, which of course is very important to our patients. Now here, delving a little bit deeper, this relatively recent study calculated that out of the injected dose of a nanoparticle formulation, roughly 7% makes it to the tumor. The rest is taken up by macrophages or degraded, but only 0.0014% actually makes it into cancer cells. The same group, Wilhelm, a couple years earlier, had predicted, based on a study that they did, comparing if a drug is encapsulated into a nanoparticle, for instance, in a polymer or a liposome or a mesoporous particle, or if it's just attached to the surface of the particle, how much signal would be received, because this particular study was focused primarily on imaging agents delivered by nanoparticles, not therapeutic agents. But nevertheless, of course, we get much greater signal or much greater therapeutic effect, we would assume, if we have a higher loading efficiency. These plots show what the, depending on the delivering efficiency, 1%, 5%, or 10%, how many injected nanoparticles would have to be delivered to reach an IC50 therapeutic response. And so it shows that this could be done with a 1% ID, 1% injected dose reaching the tumor. If 100, for the maximum delivery injection of nanoparticles. But if the drug is only surface coated, we would need a 5% injected dose. And here it shows what the number of particles that need to be injected versus delivery efficiency. So the DIE group also looked at in animals with induced tumors, well, actually xenograft models. If we look at the number of nanoparticles that are delivered to the overall tumor, we see a clear advantage of active targeting, that is targeting through, say, a folate or HER2 or EGR or ferritin or hyaluronic acid, different targeting moieties. But if we look at when they looked at within the tumor, very few of the particles were actually in the cancer cells themselves. Most of them went into the macrophages. The vast majority, well, actually not most of them, but more went into macrophages in the cancer. But the vast majority stayed extracellular, didn't even make it into a cell within the tumor. Still an advantage of the active targeting. And they showed for a different size of particles that it was fairly consistent. Now, if we look at the number of particles that, again, the number of particles that make it into the cancer, into different types of cells, so take this and sort of expand this part of the curve, because, removing this component, we see that, again, a non-significant effect of active targeting, although it certainly trends in that direction. But again, many more cells, many more particles in the macrophages compared to the cancer cells. And they showed this with different types of cells, different types of ligands, different types of tumors, and different sizes of particles. And something that I am working on with Professor Orlande is, and this is from the same group. They looked at the transport, how deep the particles go into the tumor. And not surprisingly, the particles don't make it very far into the tumor because they're associated with cancer cells and macrophages. So this was a study that they did looking at the distribution of either free drug or drug delivery in nanoparticles in a simulated tumor of a matrigel complex. And they looked at different parameters like the size of the particle, whether there's a protein corona around the particle, if there was a ligand. They looked at different size particles. They looked at different ligands, and it was pretty consistent. What Professor Orlande and I are doing is applying some models to this to try to get a better understanding of what's happening, because we have to understand not only the diffusion of the drug into the matrigel, but also its interaction with the cells. So returning to this particular slide that I showed before, only 0.0014% is going into the cancer. And that's because, again, the highly complex nature of the tumor itself, and that the particles have to get out of the blood vessel. They have to get into the tumor. They have to distribute through the tumor. And they're going to see a lot of macrophages, and macrophages which tend to be closer to the vessel. And as it turns out, these macrophages have as high attraction to the nanoparticles as the cancer cells have. Now, this Chen group did a meta-analysis of multiple studies, again, of these nanoparticles, primarily intended for imaging, not for therapy. And they showed that over the years, over 13 years of research and development, there really was no improvement in the ID, the percent injected dose that made it to the tumor. There was really no difference between passive and active targeting, inorganic or organic molecules. I'm not going to go through all of this, but these papers that I am showing slides from, these 3 papers, very interesting, and I encourage anyone who has interest in this field to get these papers and look at them. So basically what it's showing is that while there's wide variability in many of these studies, we've not really been able to pass that 1% ID mark. That, again, that doesn't mean that that these haven't been successful. Many of these are successful. They do show that they can treat the tumor, at least in animals. But the problem is that the same level of response is not translated to humans. And the response has primarily not been in extended life, but in reduced toxicities. Now, I also did a meta-analysis, but looking at particularly mesoporous silica nanoparticles and in tumor-bearing mice carrying chemotherapy agents. And what I showed is if I look at all the studies for the mesoporous nanoparticle formulations that carry doxorubicin, but not other drugs, we see a very wide distribution between the loading capacity, that is the amount of drug that can be loaded into the nanoparticle, a huge range, zeta potential, a huge range. And before I talked about the path between the particle takes between getting the vasculature and into the tumor. So the characteristics of the particle in the plasma are such that they should be neutrally charged so that they don't experience opsonization and get removed by the macrophages. But once they get into the tissue, the characteristics of the particles change. When they interact with a cell, they should have a positive charge because the negative charge inhibits the association of the nanoparticles with the cell. So the zeta potential is very important. Obviously, the size is very important also. Now, I split up the papers and looked at what the tumor inhibition ratio, that is the volume of the tumor or the size of the tumor at the end of the study, comparing no therapy to the therapy. And so that gives us the inhibition ratio. We see a wide disparity in just doxorubicin by itself. In some studies, doxorubicin had a very good, or the animals had very good response to the doxorubicin. The mesoporous nanoparticles carrying doxorubicin, a wide array. We see a improvement when the mesoporous silica nanoparticles carrying the dox is targeted. We see even an additional improvement if it is targeted, and also we include a thermotherapy or additional therapies, photodynamic therapy or radiation therapy, but they're really not much different than the liposomal dox or Abraxane, two clinically approved products that have been on the market for many years. But even in those, there's a wide variability in these animal studies. So this is the same plot, really, just showing the 95% confidence interval, showing the improvement in the targeting with the thermotherapy or photodynamic therapy, which might be better than the liposomes or the Abraxane, but there's a lot of overlap between the standard deviation, or excuse me, in this case, the 95% confidence interval. And one issue is the release rate. So we've got an issue of the drug actually making it to its site of action, but the other issue is whether or not the drug is actually being released. So for these mesoporous nanoparticle formulations, the release of doxorubicin is generally low, at least in normal pH, which is good in the sense that we don't want premature release in the plasma, which is roughly at a pH of 7.4. And then the idea is that when it reaches the tumor, and most of these particles get taken up through endocytosis, when it makes it into the tumor, the release is faster. But still, there's a huge distribution. If they make it into the tumor and we stimulate the tumor with heat, or maybe it's stimulated because of the thiol conjugation, so they're released due to glutathione in the cells, we get a much faster release. But remember, a very few percentage of the particles are actually making it to the cell. So if they don't make it to the cell and they don't get endocytosed into the cell, they're not going to be at this low 5.5 pH. They might be at a lower pH inside the tumor environment, but not at the very low pH that would be experienced by lysosomes inside the cell. So one of the common thinking is that we can improve the therapeutic effect because these particles are pH sensitive, releasing their drug, and the cancer cells have a low pH. But again, if they're not making it to the cancer cell, you're not going to have that effect. So what another one of my graduate students did is some very nice experiments looking at the transport of nanoparticles into tumors. And with help from Professor Orlande, we were able to, with my student, develop some mathematical models to predict the uptake. And here I show the experiment in cells, in cell culture, for two different types of mesoporous nanoparticles, a methoxy PEG silane and a carboxy PEG silane. Of course, the PEG is generally considered necessary for evading the opsonization in the blood and making the particles stealth. We see a big difference in the cell uptake between the methoxy particles, much greater uptake with the carboxy particles, because the methoxy particles are completely neutral. The carboxy particles have some charge to them, which allow them to interact with the cell membrane. We also see really no difference between a normal temperature, 37 degrees and 43 degrees, compared to the carboxy PEG. We see an increase in the uptake into the cells due to the hyperthermia. So the importance of this study is twofold. One, to demonstrate the, which has been demonstrated by other groups, the dependence of uptake into the cells on the surface functionalization of the particles. Also, whether or not we can increase uptake due to hyperthermia. And from these studies, we get some of the parameters that we need to further model in the three-dimensional spheroid. So here we show uptake into a three-dimensional spheroid. These are confocal images of slices from the surface, 20 microns down from the surface of the nanoparticle, 30 microns, 40 microns, towards the center of the spheroid. These are 300 micron diameter spheroids. And at 37 degrees, we see the particles primarily associated around the outside rim of the nanoparticles. So they're not really diffusing into the spheroid. Whereas if we put these same spheroids in an incubator and expose them to 43 degrees, when we add the drug, we see a much more homogeneous distribution. The particles are able to penetrate deeper into the tissue, demonstrating the advantage of hyperthermia, at least on the distribution of the nanoparticles into the tumor. Of course, we still have the issue, which we didn't look at here, was exactly where these particles are. Are they inside the cell? Are they outside of the cell? Now, these don't have macrophages. These spheroids are only cancer cells, so they don't have the macrophages. But a next step will be then to add macrophages and other types of cells into the spheroid to make it more representative of an actual tumor. So we took the parameters for the interaction of the nanoparticles with the cell and use the Arrhenius model to model the effect of thermal damage on the cells, and predicted its effect on a change in the porosity of the extracellular fluid between the cells in the spheroid. So we modeled the heat generation and thermal damage, calculate how that could change porosity and cell uptake. Then we used Fick's law to equation with reaction terms and iteratively optimized, the model to derive the optimal characteristics, the optimal parameters that govern the diffusion of these nanoparticles through the extracellular matrix and interaction with the cell and uptake into the cell. And so what we show is that if we show data from the fluorescent nanoparticles between the surface of the spheroid towards the center of the spheroid for 37 degrees and 43 degrees, we see a greater leveling out of the distribution of the nanoparticles into the center of the spheroid at the 43 degrees. And we see a very good fit between our model and our actual data. Then we also looked at comparing the transport of 50 nanometer gold nanoparticles through our spheroids. And we compared, we compared our data of our carboxypegylated silicon nanoparticles, which are about 50 nanometers in diameter. I should have mentioned that before, to what was presented by Jin et al. In 2009. And we see that our nanoparticles are able to distribute more evenly throughout the tumor. Now, of course, this isn't really a one-to-one comparison because we're comparing our data to historical data. Okay, so my last three slides here, I'm pretty much finished. What are the major clinical challenges? Well, clearly the difference between animal models and human disease, finding the optimal design for each different cancer type. The pharmacokinetics, the overall pharmacokinetics. I talked about the fact that the characteristics of the nanoparticles, really the optimal characteristics are different between when the at the absorption site or in the blood all the way down to the cells. And also, what clinical endpoints justify the added cost compared to traditional treatments? Are we looking for a complete response, overall survival, progression-free survival, time to progression, or reduced side effects? And what would constitute our silver bullet? Our silver bullet, would we have to have complete response? Or, what, even if we did get overall survival, how much would we really be looking for? And then there are challenges to commercialization, the difficulty to scale up production and control these physical properties. And of course, the cost of this production, poorly defined regulatory process at least in the United States, and I assume in Brazil and in South America, similar. They don't really know how to handle these types of drug formulations. The intellectual property, of course, companies aren't going to go through the trouble of developing these so that they actually can be administered during the clinical trials if they can't own they can't make money. Again, the difference between preclinical models and human disease. No standardized reporting of methods, which is what I made a big deal about in the meta-analysis paper that I published. There's so much variability in how data are reported that it's really hard to combine the studies together to make any sense out of these hundreds and hundreds of papers that are being generated. Very complicated tumor microenvironment, the barriers. EPR is not ubiquitous, and it really has been questioned of how important EPR really is in this process. So I have some recommendations. I'm not going to go over this slide. If you're watching this recorded, you can pause the slide to see. Going back to this point here of no standardized reporting of methods in results, many of these are missing in most of the papers that I reviewed, and I reviewed about 200 papers overall of Mesoporous silica nanoparticles delivered to tumor-bearing mice. And just the fact that many of these data are not reported or the data are confusing makes it very difficult to come to some kind of conclusion so that we can start looking at all of these 100 different formulations that are presented. And those are just the formulations that were tested in animals, in a tumor-bearing animal where they showed therapeutic response. There are many, many more papers where they're limited to in vitro studies, which I didn't include. There are hundreds and hundreds. But again, how do we come up to an agreement, or how does a company say, wow, I want to take one of these formulations, I see potential in them, and moving it forward to product? And so recommended characteristics that really need to be reported. And many of these are either not reported or it's somewhat confusing and not really clear. So that's what I have to present. Hopefully I have convinced you that that nanoparticle drug delivery is still a very useful approach to be considered, that there has been improvements in the commercial products that have been, the products that have been commercialized. There's been advantage to the patients, obvious advantage to the companies, but we need to do better. And with all of the hundreds of papers that are presented each year on new formulations, how do we dig through those data to try to stop the madness, so to speak, and really kind of focus down on what could be potentially a silver bullet? And for this audience, we still have a wide gap in our understanding of the basic oncobiology to understand, is there really, the theoretical advantage of nanoparticles and hyperthermia therapy that we've been writing in the papers for 20 or 30 years, are they real? What is the real oncobiological foundation for that? So again, thank you very much for your attention.
Okay.
If we have time for questions,
Thank you so
happy
much,
to,
Anthony, for the excellent presentation. So yes, we have time for questions. Alguém quer colocar as perguntas no chat, abrir o microfone, como vocês quiserem. So my first question, make my own question. So what do you think about finding new targets? As you have shown, this slide is
Right,
just
right.
perfect. So if you say that nanoparticles are better to reach macrophages and the cellular matrix, so how do you think about looking for new targets? I mean, macrophages or the ECM itself?
Exactly, right. Very good question. And I think that's really where we need to be focusing more, because unless we can figure out how to get more into the cancer cells, that's what we need to get at. Now, again, I'm an engineer, so I leave those kind of important questions to you all, but it's up to your group to identify the targets. And then it's up to people like me and Professor Orlando to try to design the vehicles to get to those targets.
Perfect.
And targeting macrophages is probably the next step. And also delivering different types of drugs. And for instance, immunological, looking at immunotherapy instead of chemotherapy is also, I think, very exciting.
Exactly. And also other cells in the tumor microenvironments, maybe.
I'm sorry.
Other cells from the tumor microenvironments.
Oh,
maybe
yeah, right.
Also another
exactly. There's tons and tons of research has been done in looking at the tumor microenvironment, but tumors are very heterogeneous, right? And so identifying what is the tumor microenvironment of a particular cancer in a particular patient is a real trick. So we need theranostics along with the of these approaches so that we can identify the patients that are more likely to respond to these, expensive and complicated therapies. So if we can learn what the target should be, then and what therapeutics should be delivered, how fast they should be delivered, at what concentrations they need to be delivered, whether it should be time released, and what combinations. That's what we need to then go back to our engineering and try to come up with a solution for you.
So we have some questions here. Actually, one person asked, could the intratumoral mass injection of nanoparticles rather than IV improve delivery to cancer cells? Although it's less interesting clinically speaking.
Right. It definitely improves. It definitely improves therapeutic outcomes. And in my analysis that I did, my meta-analysis, I specifically excluded particles that were injected directly into the tumor. So I only looked at intravascularly injected drug. And the reason I did that partly is because there are many more of them, and not to confuse, you can't really compare the two, but for sure you get a higher response. But the problem is you're not going to treat the metastatic disease.
Yes, sure. Another question, could you talk more about the stability of your nanoparticles? Concerning PEG, it was easier to make it stable for biological assays. So
I think that, so the question is the stability of the nanoparticles. When we talk about stability, we talk about two different things. Well, we can talk about stability in terms of how well they're able to stay in suspension without aggregating and sticking together. And that's where the neutral charge comes into play, because actually a neutral charge will actually cause the particles to want to stick together because there's no charge to keep them repelled. But the polyethylene glycol, even though it's a neutral charge, it has sort of arms and brushes, actually, it's referred to, that keep the particles apart. But if we're talking about gold particles or iron oxide particles, they're going to want to stick together. So that's one part of stability we talk about. Another aspect of stability is whether or not they degrade inside the vasculature as they're being delivered through the bloodstream. And the liposomes are much less stable. They can more easily be degraded than other materials. Now, polymer materials have the advantage that they do degrade, but their degradation is not very controllable. Mesoporous silica nanoparticles, one disadvantage is that they degrade very slowly. Now they do degrade into silica, which is considered, non-toxic, generally acceptable material, but they degrade over a long time.
Okay, so we have another question and a comment. So from Dr. Claudia Lage, I do agree that the silver bullet to fight cancer will count on the combination of nanoparticle delivery drugs with biophysical treatments, as hyperthermia or electric fields applied in situ. No, do you any comments on that?
I'm sorry, I didn't.
So that's the comment made by Dr. Claudia Lage, that she agrees that the silver bullet to fight cancer will count on the combination of nanoparticle delivery drugs with biophysical treatments, so such as hyperthermia or electrical fields. So, okay,
Well,
I'll skip.
Yeah, I agree. I think that theranostics, as I said, to identify patients that are likely to respond, and combined therapies. I don't think that nanoparticles will ever be a first-line therapy on its own. But then I'm sure this audience knows that surgery of a single solitary, well, it doesn't have to be a single, but solitary tumors is really the only cure. Thyroid cancer is somewhat curable, but most cancers, you all know, are diagnosed, tend to be late stage, metastatic. And so surgery is not a cure option. The silver bullet is intended, the idea of a silver bullet, which I'm sure, hopefully you'll understand the silver bullet kills the werewolf. That's the only thing that can kill a werewolf, right? But at any rate, it depends on recognition and specificity. So I didn't really talk too much about specificity, but that's really the key. Is specificity, and the nanoparticles are intended to make them more specific, either because of the enhanced permeability and retention, or some type of targeting, or a targeted hyperthermia, something like that. So I agree, yes.
Yeah, so we have perhaps the last comment. So for Dr. Gabriela Nestal, so thank you very much for the great talk. It seems that there are many challenges for targeting solid tumors with nano formulations. I wonder whether hematological malignancies would skip at least some of these challenges or would face additional ones. Do you know how nanotechnological therapeutics are being approached for leukemias?
Yes. There are several successful applications in leukemia. So fluid cancer, because the we can take advantage. That's a nice marker right there for almost a natural marker, right, for these nanoparticles to be taking up into liquid cancers. So that's a definitely a area of interest. People are looking at it, and I think that has a lot of potential. When we talk about tumors, the tumor model that I was showing in these spheroids, they're avascular. they're about 300 microns in diameter, which is about the limit of size before when they start generating a hypoxic core, because oxygen and nutrients can't get to the center of the spheroid. So treating larger and larger tumors becomes difficult because the interstitial pressure of these tumors builds up, and it inhibits the introduction of nanoparticles into the tumor itself. I'm not sure if that answered the question.
I think, yeah, perhaps by facing other cancer types that do not that are not related with those constitution of microenvironments, and then so I guess hematological malignancies, as they are circulating cells,
exactly,
except like that ones that are in the bone marrow.
Right, because then you don't have the issue of then you don't have the issue of the EPR and the distribution through the tumor microenvironment, that sort of thing. Yeah.
Okay, so we don't have more comments here, and so Anthony, I would like to thank you so much for the excellent talk here. It was a pleasure receiving you here today, and I sent you the comments, the chats. I have just saved them, so I send you by mail, and I hope we can hear you again in the near future. And thank you, Elcio, for inviting Dr. Anthony to talk to us today. So thank you so much.
All right, thank you all. I really appreciate it, and I hope to make it down there sometime when all this pandemic mess clears up, and it'd be nice to visit. All right, bye-bye.
I hope so.
Thank you, Anthony. Thank you, Anthony.
Bye-bye.
Great talk. Thank you. Thank you, Robson.